Welcome to Department of Botany, Serampur Girls College, myself Dr. Madhusri Haldar. Today our topic of discussion is Pteridophyte, and today we discussed about the life cycle of one Lycophyta member, that is Selaginella. First of all, we have to know the habit and habitat of Selaginella. Here that is the morphological picture of sporophytic plant body of Selaginella. Selaginella shows considerable variation in size, symmetry, and morphology. Mostly they are herbaceous, perennial, however, a few are annual also. Majority of Selaginella dorsiventral, prostrate, and creeping on the surface. For example, Selaginella kraussiana, and some are radial and grow erect. For example, Selaginella selaginoides, and a few are climber also. Most of the species of Selaginella grow on the ground in humid and shady habitats. A few species are xerophytic and grow in arid condition on exposed rock surface. On the basis of the habitat preference of Selaginella, some species are known as resurrection plant. There are some xerophytic species of Selaginella whose rolls up into compact ball of seemingly dead leaves and stems during the dry season, but unrolls to carry the normal activities when put in water. Such plants are commonly known as resurrection or Mrit Sanjeevani plant. Next thing we have to discuss about is the salient features of Selaginella. First is the sporophytic plant body is herbaceous and the shoot is either dorsiventral or radial, either creeping or erect. Next, the leaves are small, that is microphyllous leaves are found, and unique character is ligule present at the base of each leaf and sporophyll. Next, number 3 is rhizophore. Rhizophore is actually a leafless structure where from root arises. Next, sporophylls are usually aggregated into strobilus at the apex of the branch, and Selaginella are heterosporous. And heterothallic also, that is the plant body is dioecious. Next, antherozoids are biciliate. That is the morphological diagram of Selaginella sporophytic plant body. Next thing is Hieronymus 1902 divided the genus Selaginella into two subgenera. That is, one is Homophylum and another one is Heterophylum on the basis of general structure of the plant body. In that case, the two subgenera are Homophylum and Heterophylum. Homophylum bears foliage leaves that are more or less uniform in size, that is the isophylous leaves, and in case of Heterophylum, foliage leaves usually are of two types. That's why they called it as heterophylous. In case of Homophylum, arrangement of leaves are spiral, and in case of Heterophylum, arrangement of leaves are into 4 vertical rows, 2 vertical with large leaves and 2 dorsal with small leaves. Next point is two ventral rows with large leaves and 2 dorsal rows with small leaves. Next point is plant body is erect and dichotomously branched in case of Homophylum, and in case of Heterophylum, plant body is prostrate and dorsiventral. In case of Homophylum, there are only 50 genus and 50 species actually, and in case of Heterophylum, there are many species. As example, Selaginella kraussiana and Selaginella helvetica. Next is morphology of rhizophore of Selaginella. In the word rhizophore, there are actually two words, rhiza and phora. Rhizo means root and phora means bearer. In many species of Selaginella, peculiar leafless prop-like cylindrical structure originated from the stem at the point of branching. These grow downwards into the surface and form many adventitious roots at their free ends. They are known as rhizophores. The morphological nature of rhizophore is actually very controversial because of its unusual position and structure. And there are actually many controversial view of scientists that it is a root or stem or an organ sui generis. That is an organ neither a stem nor a root. So we have to know the similarity of the rhizophore with the root. The first similarity is rhizophore are positively geotropic in nature as we shows in root. It does not bear leaves. Monarch xylem like that of root. Presence of root cap in some species like Selaginella densa and Selaginella kraussiana. That is a report by Ochok and Steves in 1967. Next is transport of auxin in rhizophore is acropetal, which is similar to that of root. That is also reported by Ochok and Sensex in 1974. Similarities with stems. Firstly, rhizophore is exogenous in origin like that of stem. Absence of root caps and root hairs. Originate due to activities of meristem which are present between two branches of stem. This meristem has been termed angle meristem, which is basically an embryonic shoot, and the production of roots exogenously from the tip of rhizophore. And lastly, under experimental condition, rhizophore can be transformed into a leafy short, that is reported by Gifford and Foster in the year 1959. So there are many controversial view and controversial thing, but Bauer and Goebel suggested that rhizophore is neither a stem nor a root but an organ sui generis. According to Scott 1938, it is a specialized stem, behaves like a root. But recent biochemical studies from the protein extraction of stem, leaf and rhizophore revealed the fact that the polypeptides of rhizophore more closely resemble those of stem rather than subterranean root of Selaginella. Next important thing, we have to know the structure of ligule. So what is ligule? A small membranous tongue-like structure is located at the base of each vegetative leaf and sporophyll. That is the diagrammatic view of Selaginella. Actually, the ligule has a basal cup-shaped sheath with often Caspadian strips and an adjacent hemispherical glossopodium. Glossopodium is a Greek word. Glossa means tongue and podium means foot. Next, the function of ligule. Number 1 is ligule act as a water absorbing structure which keeps the young leaf primordium and sporangium moist during the early development. Next is in some species during early development, ligule secretes a mucilaginous substance composed of carbohydrates and proteins that keep the apical meristem moist and prevent them from desiccation. Another most important thing comes after ligule is the stem. Morphologically, the stem is generally erect or radial. In some species, they are prostrate and dorsiventral with lateral branching. But the important thing is the anatomy of the stem. The anatomy of the stem of mature Selaginella is very distinct and differentiated into an outer epidermis, middle cortex, and centrally located stele. Outer cell walls of the epidermis is cutinized and stomata absent. Cortex is usually made up of compactly arranged parenchymatous cell without intercellular space. In most species, it is comprised of two layers: outer thick-walled sclerenchymatous cells, that is hypodermis, and the inner thin-walled parenchymatous cell, while the cortex of maximum xerophytic species comprised of sclerenchymatous cell. Next thing is the trabeculae. The cortex is actually connected with the centrally located stele with the help of some elongated, radially elongated cells known as trabeculae, and actually the trabeculae has the Casparian strips. And there is much variation in the vascular cylinder among the different species of Selaginella. These are the variations. The number of stele may differ from 1 to 16, and that varies from species to species, sometimes in the same species also. Actually, this is due to the desiccation, but young plants invariably shows monostelic configuration. One polystelic species also found, which contains 16 stele, that is the Selaginella levigata. That is the diagrammatic view of transverse section of Selaginella stem, that is a monostelic condition, and that is the diagrammatic view of Selaginella stem with two stele, that is distelic condition. Basically, stele is protostelic type. Xylem is surrounded by phloem and no pith is there. Each stele composed of pericycle, xylem, and phloem. Pericycle composed of thin-walled parenchymatous cell. Xylem is exarch with one or two protoxylem groups, maximum are metaxylem. It consists of only tracheids. Phloem consists of sieve cells and parenchyma. Companion cells and trachea absent. Next important thing is reproduction of Selaginella. Sporophytic plant body of Selaginella reproduces either by vegetative and sexual method. In case of vegetative reproduction, Selaginella use a tuber or dormant bud and through bulbils, but our interest is the reproduction by spores or sexual reproduction. One thing comes in our mind, that is the strobilus. What is strobilus? Numerous haploid spores are produced in sporangium, plural sporangia. Sporangia are located in the axil of a leaf-like structure called sporophylls. The sporangia and sporophylls together spirally and compactly arranged to form a cone or strobilus, plural strobili. That is the sporophytic plant body which bears strobili, and that is the cross section or transverse section of one single strobilus. Each sporophyll has ligule at its base and a stalked sporangium on the adaxial surface. Thus, the sporangium is present between ligule and the main axis. There are two types of sporophyll. One is microsporophyll and another one is megasporophyll. Now, sporangium and spores. Sporophyll are of two types. We already know that. Sporophyll bearing microsporangia called microsporophyll, while the megasporophyll bear megasporangia. Let learn about the microsporangium and the megasporangium. Microsporangium is rounded or ovoid in shape and red or brown in color. Contain numerous microspore. Microspore are circular in shape, while megasporangium or macrosporangium is four-lobed structure. The color is green, actually green is white or orange in color but pale, contain only 4 megaspore. Megaspores are tetrahedral in shape. That is the picture of megasporangium of Selaginella, and that is microsporangium. Both type of sporangia are stalked structure surrounded by sporangial wall consisting of two outer jacket layers and one inner tapetum. Outermost layer of jacket is thick and columnar and its cell contain chloroplast. Inner layer consists of thin-walled elongated cell. In majority of cases like Selaginella kraussiana, two lowermost sporangia are megasporangia, all other are microsporangia. While in Selaginella liniculifolia, one side of strobulus contain microsporangia, another side has megasporangia. That is the view of the Selaginella liniculifolia, which bear microsporangia in the left side and megasporangia in the right side. While in the left picture, that is the Selaginella kraussiana, lowermost sporangia is microsporangia and others are microsporangia. Next is the life cycle of Selaginella. Before that, we have a brief idea about the life cycle of Pteridophyte. Here, the sporophytic plant body is diploid or twin, which bear megasporangium and microsporangium that produce megaspores and microspores. And from that megaspore, female gametophytes are produced, and from microspore, male gametophytes are produced. Female gametophyte produce archegonium within it, and male gametophyte produce antheridium or male structure within it. Antheridium produce antherozoid, archegonium produce egg by meiosis division, that is the n number of chromosome. After fertilization, zygote is produced from antherozoid and egg. So, zygote is diploid, embryo is developed from zygote, that is also diploid, and from embryo young sporophyte is developed. So, there are two distinct phase or alternation of generation, that is the haploid phase and diploid phase. Now come to the portion of life cycle of Selaginella. That is the sporophytic plant body of Selaginella, that is the strobulus, which bear both mega and microsporangia with mega and microspores that produce microsporangium, that is 2n or diploid, and megasporangium, which bear megaspores, that is also diploid. Microsporangium produce microspore mother cell, that is also diploid, from which microspore are produced by meiosis division and the chromosome number is n. From microspore, male gametophytes are produced and male gametophyte produce sperm, while in case of megasporangium, they produce megaspore mother cell which have diploid chromosome number. Megaspores are produced by reductional division and the chromosome number is n. From megaspore, female gametophyte is produced and female gametophyte bear archegonium which contain egg. After fertilization of egg and sperm cells, zygote is formed which is diploid or 2n. Young sporophyte is developed from zygote, and this is the whole structure of Selaginella life cycle. And in this life cycle there are two distinct phases. One is sporophytic phase or 2n diploid phase, and another one is gametophytic phase or haploid phase. That is the whole life cycle of Selaginella, which shows distinct alteration of generation. That is all about the Selaginella life cycle. And next thing is seed habit of Selaginella. That is, Selaginella actually shows some characters of seed habits. By evolutionary pathway, heterospory condition arises from homosporous condition. Heterospory means two types of spore. Homosporous means all spores are alike. And from heterospory, seeds are evolved. Selaginella shows traits which resembles with characteristic essential for formation of seeds. These are as follows. Number 1, heterospory, that is megaspore and microspores are found in Selaginella. Number 2 is number of megaspores reduced to 1 in Selaginella monospora. Number 3 is gametophytes are nutritionally dependent on parent sporophyte, that is male and female gametophyte. And next thing is in Selaginella rupestris, megaspores never came out, and all the processes like fertilization, zygote formation and development of rhizophore etc. takes place inside the megasporangium. This shows similarity with viviparous germination of angiosperms. So, on the basis of above mentioned features of Selaginella, it can be concluded that Selaginella has some characteristics of seed plant, but it has not achieved the seed habit fully. So, that is all about Selaginella. Thank you.